Today I want to tell you a little bit about, this is an organoid session, so let me begin with, I think, an organoid which I think needs a lot more development and applications in our, especially in preclinical and pharmaceutical sort of intervention testing, is I'm going to talk to you about human 3D skeletal muscle organoids. And I'm going to tell you a little bit in the beginning about what the need is, where we are in the lab in terms of developing such a model, and then a few applications that we are moving forward. So first of all, I don't have to tell the audience here what an important role skeletal muscle plays. Of course, in the context of lots of diseases, muscular dystrophies and other genetic diseases play a major need here in terms of treatments. But of course, muscle does a lot more than just mechanical work. It's also really plays an important role in metabolic disease. It's actually the site of major energy metabolism and heat generation and so on. And so no wonder that muscle dysfunction, muscle mass, muscle function plays an important role in aging and how aging essentially manifests itself in sarcopenia. And of course, we won't go into the other aspects in terms of disuse, atrophy, and due to inactivity. Of course, there is also a lot of skeletal muscle loss, which can play a major role in recovery. I won't talk to you about today also about some exciting areas we're studying on in terms of effects of spaceflight on microgravity, on skeletal muscle mass. And of course, we think there's an impending, if you will, a whole bunch of people coming in who are taking GLP-1 therapies at this point. And one of the well-known side effects, if you will, is that as effective as muscle loss or weight loss is in these kinds of patients, there is also a disproportionate loss of skeletal muscle mass. So we do think that there is going to be an impending need for understanding skeletal muscle atrophy in multiple contexts. So there is, of course, an even more, I think, an urgent need for us as a scientific community, for our country certainly, to start developing organoids as a model for disease and for pharmaceutical testing. As many of us know, that the writings on the wall and the landscape is rapidly changing, that in terms of either understanding disease etiology or in terms of looking at drug efficacy or toxicity, there is a real need now to move towards organoids, integrating them to our workflows for studying drug discovery. As you know, the FDA 2.0 Modernization Act essentially says, though animal, of course, testing is not going away anytime soon, it's really a tectonic shift in that no longer is animal testing required in showing regulators that the drug is toxic or efficacious. So I think along with AI and other kinds of virtual testing of drug efficacies and all of that, the field is rapidly moving into looking at alternative non-animal models for looking at drugs and disease efficacy. So that also prepares the landscape for looking at multiple organoids, and my lab, of course, focuses a lot on skeletal muscle. So I just want to spend the next few slides just setting up the notion that though, be it for dystrophies or be it for many of the diseases that we look at, there are very well-established animal models, and they have performed well in terms of giving us some insights into at least basic mechanisms of disease. They don't necessarily serve the function of looking at disease etiology in terms of when the disease actually onsets and how drugs actually affect different tissues and different species. For example, I'll just in the next couple of slides just give you some basic highlights of what I think are the major changes between rodents and other models and humans. For example, though it's the basic structure of skeletal muscle in terms of the sarcoplasmic reticulum, the thin and thick filaments, and the contractile units are conserved between number of animals. There is some key features which are very different. In terms of the distance between the sarcoplasmic reticulums can be a bit different. The size of the NMJ clusters and the ACHR clusters are very different. And this really manifests in terms of how functionally these muscles are different, which is what's important. So in the normal rodent models that are used for a number of diseases, we know that smaller animals are predominantly fast-twitch type 2 fibers. And in human beings, that is very, very different. We have a lot more slow-twitch type 1 fibers. So in terms of looking at how metabolism and how drug efficacy manifests in metabolic disease, it certainly requires that we have more complex fiber models for studying these. Same thing goes for, so as you can imagine, the contraction speeds are very, very different in terms of fast twitch fibers and slow twitch fibers. The calcium handling in terms of calcium release kinetics and how they respond to different agonists are very different. So you can also then from this also extrapolate to the fact that the metabolism is very different. So essentially the glycolytic fibers, the glycolytic enzyme activities in smaller rodents are extremely high, while we have a lot more of the oxidative fibers and mitochondrial fibers in human beings. So there are a number of other differences, but just want to some salient points as to why we need to move towards organoids that are much, much closer to what we as human beings have. And even functionally, of course, I thought it was worth putting this slide as well, that if you look at the kind of muscles that are used by different animals, they are very, very different. As you can tell, that this is just looking at muscle excursion and looking at the amount of muscle stretch, if you will, as human beings ambulate versus a small animal like a mouse. Most of our work and most of our load bearing is done by, you can see the red bar essentially tells you the muscles that are being activated mostly in human beings versus those in smaller animals. You can tell that the vastus muscles and those in the upper limb are much more load bearing and do a lot more work as compared to the soleus muscle and so on in rodents. So it really, though we have learned a lot in terms of skeletal muscle physiology and function using small animals, it's really time to move to making muscles from human beings to really understand how the things manifest there. So in the next few slides, I'll very quickly just tell you how we have developed this model and a couple of applications that we are rapidly moving towards. So the lab essentially is interested in understanding the aging context of human beings. Therefore, we have, at this point, though we are beginning to work with iPSCs and iPSC-derived myoblasts, we have been using myoblasts from biopsies from human quadriceps and isolating myoblasts from that. So essentially, it's very, very simple. The idea is very simple. To actually form muscle fibers, you need tension, you need mechanical stress. And the way we give that kind of mechanical stress is by making a small 96 well-sized PDMS scaffold, which have two posts embedded within them. And these posts, if you will, perform the function of a tendon. Around it, a muscle is formed, and over time, it organizes itself into 3D human myofibers, and I'll show you some histological evidence to suggest what kind of muscles they are, and also some functional data that we're getting. So essentially, its idea is really simple. In some hydrogels with some atrogel and growth factors, we put the myoblasts embedded in those hydrogels into the scaffolds, and they self-organize into these, over about 12 to 14 days, they start organizing into these myofibers. And how do we know that they are myofibers? This is essentially a stitched image of entire myofiber, and it's about 1.5 millimeters, a few millimeters long, depending on the kind of scaffolds we use. But so we were gratified in one case to see that after 12 to 14 days, you can see aligned myofibers that have myosin heavy chain, and I'll show you the fast and slow twitch sort of stains that we are also beginning to get. Also happy to see that there are these acetylcholine receptors that are aligned across these fibers, also telling you that maybe these fibers are ready for some sort of a functional stimuli. Next, one of the things that you do not get in 2D systems and so on is actually the formation of these beautiful sarcomeres that are important for releasing calcium and helping the contraction of the muscle. So we can see that there are these well-aligned sarcomeres that start arising at day 14. And the sarcomeric distance is also very similar to what you would expect in a human being, except different from a mouse. Now, to our knowledge, this is one of the more mature fibers that we have been able to generate. So we have the myosin MYH and the MYH2. I was telling you about humans having both slow and fast twitch fibers. And now, over time, we are beginning to see effectin essentially just stains all the fibers. So this is a cross-sectional section, as you can imagine, and this is a longitudinal section. So the cross-sectional side, you can see that there are multiple fibers inside the muscle organoid, and we can also see there are multiple fibers, and they're maturing into both slow and fast twitch. And to our knowledge, this is one of the more mature fibers that we have seen in literature. It's going beyond the embryonic myosin heavy chain stains. So this told us that maybe these organoids are mature at 14 and 15. Now we have, due to some needs, have gone up to even day 40 and so on, and we haven't really analyzed it. Because of the lack of time, we usually stop these experiments at 15 and 16. That's when we do the analysis. But I expect that given more time, there might be more maturation and changes in these fibers, and we hope to look at that in the near future. So then, now that we can see that these fibers are, aligned, and you have ACHR units there, next thing we want to know whether it can respond to acetylcholine. And I don't know if somebody can help me. Hold on. Yeah. So at 10 seconds, we add acetylcholine, and you can see that the calcium is taken up by muscle, and hopefully you can also see that the muscle actually contracts and moves in a quite a recognizable sort of way. And we can quantify those things. You can see those at the end, the graphs, which essentially quantify the amount of calcium that is being taken up after addition of acetylcholine, and also calculating through microscopy the displacement of these cells. And we are beginning to see now some force production, response to acetylcholine, and now based on the Young's modulus of the PDMS and the movement of the cells, we can actually start calculating the millinewton forces that are being produced. So what we are hoping now is that we have a maturing human 3D skeletal muscle organoid that we can start looking at effects of diseases or drugs on functional outputs and as well as histological outputs. So there are a couple of problems we are beginning to ask with this. We work with this company called Mitopower, and there is of course a huge amount of interest in NAD supplements and aging and all of that. And the NAD supplement we are using right now is this compound that's called NRH. Just wanted to tell you there are multiple ways of enhancing NAD levels in cells, and there seems to be a notion in the field with some good evidence that NAD levels, possibly through sirtuins and possibly through other metabolic mechanisms, can drive better aging outcomes and so on. And there is, I think, an unfortunate huge push in this area that people off the shelf are beginning to take a lot of these NAD supplements. I know many of them. And I think it is really important that in a human context, we really need to rapidly start studying what these compounds actually do. Are they toxic? Are they beneficial to muscle? Because the evidence is very contradictory. You can read some papers and convince yourself that this is a wonderful thing to promote muscle function. And there is also a lot of contradictory evidence that suggests that either it might not be as effective as we think, or they might even be toxic. For instance, of course, there is evidence to suggest that that there's some supportive evidence, and there is evidence to suggest that supplementation with NR and in some cases some other supplements can actually inhibit muscle regeneration and in some cases force production and so on. In some cases, of course, there have lots of high-profile papers which have shown that increased NAD levels can improve aging-related sarcopenia and performance and so on. The jury is out there, and I think a lot of this confusion is mainly because we have been using small molecule rodents and maybe some 2D sort of data to get at some of the things, and I think human beings respond very differently. So we wanted to now start using these organoids to really start understanding what do these NAD compounds, what are they good at, what are they not so good at. So I'll give you some early data that we have from the lab, where essentially we started with a high dose of NRH, about 500 micromolar. Usually people use about hundreds of micromolar, but they go up to 500. So we wanted to see at the highest dose that people use, what kind of effects do we see. But we do see that there is an improved amount of, so on the right side we are calculating fusion index, and fusion index is essentially looking at all the red MYH stained and seeing how many of those have DAPI within them. So how many fused nuclei, myonuclei have been formed. So essentially we calculate a fusion index, if you will, for the entire organoid, and then we compare it with the treatment of the compound, and we can see that this NAD analog that people are thinking of using in the clinic does improve fusion index. Not a huge significant improvement in terms of the replication of myoblasts while this is happening, but that also could be a factor. We see very nicely that NRH can improve the fiber cross section. I had shown you some evidence of the cross-sectional area, and it does increase the cross-sectional area of the fibers. As such, there is no huge difference in the statistically significant difference in the formation of fast and slow muscle fibers, but that also seems to be happening. We need probably more replicates. So we saw this and we thought, great, it might actually make muscles much more functional now and bigger, and maybe people are right. But in fact, and we of course, now this is a TMRM staining of mitochondria and the fibers, and NRH treatment does increase the TMRM intensity, telling you that, yeah, the mitochondria are better aligned in these organoids, and that there is also an increase in, and we also found that there are bigger puncta of these ACH clusters that you can see below with an RH treatment. So all of those things told us that maybe this compound is actually quite beneficial. But we were quite surprised that when we start looking at the sarcoplasmic reticulum, the SAA formation, this compound actually impedes the organization of the SAA. So it's not all good. As a matter of fact, when we actually start looking at, I had shown you the calcium uptake assay. So exactly as expected with these disorganized SAA, their ability to take up calcium is completely impeded. And of course, this is not what the company wants to see, but this is the data. And I think is really, really important that when you have these sorts of natural supplements and other things that we're taking, which have shown really beneficial effects in animals in multiple contexts, it is important now to move these things into a human context to see what happens at different concentrations. So we are still working on this, and this is a manuscript that will go out very, very soon, which tells you that using these organoids, you can get into some nuanced aspects of what is beneficial and harmful about some of these supplements, and we hope this will give some pause to the supplement area of incomes of NAD. The second thing I'm not going to tell you a little much more about, this is a collaboration we have with TIGS on their ICMR grant with Professor Arvind Ramanathan there. And there we are, again, there is a large consortium of medicinal chemists and us, and of course a lot of clinical scientists and doctors who are interested in looking at GNE myopathy. So I don't have to explain to you here what GNE myopathy is. You probably have seen multiple patients here. But essentially this is you can see in that, I don't know if I have a pointer there, but you can see that over time there is a major loss of skeletal muscle just over a few years. In the teen years, many of these patients do not have very much skeletal muscle mass left. And in a histological sense, there are multiple vacuoles that are formed inside their skeletal muscles, and there are also number of amyloid sort of aggregates that are present in these myofibers. And the reason, of course, is that there is a mutation in this enzyme, which is a UDP gluconac 2 epimerase, also called mannose N-acetate kinase, acetyl kinase, and because of that, the cell is not able to produce sialic acid, and there's all kinds of problems with glycosylated proteins. Now, of course, it is thought that it is 1 to 2 patients per 100,000, which is quite prevalent in India, and there is a lot more screening and all of that going on. So what we want to do now is to use these. So the problem is that there are some mouse models available. Many of these GNE knockout mice and these mutants are not embryonically viable. And in many, many ways, the etiology of the disease is very different. Though it takes a while for human beings to manifest this, it's very rapidly present in these mice. And of course, I think because of the type of fibers and the type of load-bearing muscles there are, unlike in humans where you start encountering it in the distal part of your muscles, it is not so in mice. And there is also I was telling you about the inclusion bodies and the vacuoles and so on. Those aren't necessarily seen in these mouse models. Telling you that there is a real need to now start modeling these diseases in a human context so that now we can start looking at interventions in a more preclinical sense. And Vasant has now, one of the mutations, the ILE618THR, is apparently one of the most prevalent mutations in India. And Vasant has now created this mutation in H9 human ESC cells, and now we also have a doxycycline-inducible PAX7 model there. So we have come to the point where now we can efficiently start making myoblasts from these knockout these mutated HESCs. And now what we want to do next is we are beginning to build these human organoids to see whether we can start seeing these sorts of features in terms of myopathy, in terms of these vacuoles and these kinds of histological evidences that I'm hoping in the next few months we will start seeing. So hopefully next time I'm here I can show some data of organoids modeling this disease. So I'll leave you at that and just want to thank our amazing institute and all the support we get from BRIC-Institute for doing all of this work. Thanks. Yeah,
thank you, Dr. Ramanathan, for sticking to time. Like I said, we are a little short of time, but we can take a couple of questions.
Exercise-induced effect on these kind of organoid muscles. Like,
you're absolutely right.
PDMS allows you to stretch and de-stretch.
Yes. So now that I didn't show you that data, it is just coming in. Now we are putting electric current to make them do the twitch and tetanic and quantifying it. But soon, hopefully, we can have exercise regimens there. So we are starting there.
I see. And I'm another curious question. You mentioned about the nucleate fusion. Do you mean nucleus actually fuse?
No, sorry, it's not. It's cell fusion. So it is essentially as like our muscles, they are fibers, right? They are no longer individual cells.
Got it. Yeah.
But yes, whether nuclei fuse, I will not be surprised if nature does that, because we used to always think these are post-mitotic nuclei. Recently, somebody showed EDU incorporation there as well. So there are still some mysteries. Yeah.
I have a rather basic question. So when you talk about the aging muscle niche, it's not just cell autonomous. You have increase in adipocytes, you have macrophages, right? And the niche factors are the one which are basically causing the aging. So does your organoid model encompass these features? Because
right now, no. So I guess we should. Right now, it's just the contractile unit.
Yeah.
Now we could, in theory, incorporate other cell types in there, which we will start beginning to work on. But that said, I think there are clearly niche-dependent effects, but there are also cell-autonomous effects. And how do I know that? Because if you take old myoblasts and so on, and remove them and put them into a young niche, they are still dysfunctional.
Yeah.
So though they are much more improved, I think the niche plays a major role. It's fatty infiltration. The inflammation plays a major role in sarcopenia. Yeah. But yeah, right now we are only looking at the contractile unit autonomous effects. Yeah.
Okay, thank you.
Yeah, Arvind. So just the two questions I wanted to ask, but I think it's also important to look at the systemic physiological impacts, because otherwise you don't have endocrine inputs, you don't have neuronal inputs. There's a lot of metabolic channeling that actually feeds the muscle that is absolutely missing in the organoid system. So I think the challenge would be to build these in a layered manner, and otherwise organoids will have the same limitations as you have in mouse, because they will never recapitulate things.
Agreed.
Right? Agreed. Now, quickly going back to the NR and the NAD homeostasis, I think, do you have a sense of the basic redox differences between mixed fibers versus predominantly type 1 or type 2? Because I think that's where the big gap is. I think people are randomly choosing this without understanding the redox balance between type 1, type 2 and the mixed fibers.
You're absolutely right. I think at least when you added this, it didn't seem to there was a trend. So we didn't see a statistically different increase in the type 1 fibers, but I think it is impacting the kind of fibers that are arising from it. So the question is, I don't know right now about how they are different in different fibers, but the point of making these into more complex things is very, very well taken. Now that this is there and they're ACHR, we're actually thinking of co-culturing motor neurons and other things. So there's, of course, a certain limitation to how it could ever truly mimic what's in the body. But I think knowing the limitations, you should ask those kinds of questions. Given that this is it, what's there? But I don't think we'll truly be able to get the immune cell. There are 36 cell types in muscle, so I don't think we'll get there.